The New England Regional Spinal Cord Injury Center and Spalding Harvard Spinal Cord Injury Model System would like to welcome you all to the Knowledge in Motion webcast this evening. We would also like to welcome our international webcasters. We have people joining us from Canada, the UK, Brazil, Italy, Spain, Portugal, India, and Ethiopia, and from 31 states across the United States, and that's over 400 people that we have, in addition to all the people that we have in this room at this lovely new Spalding facility that's really amazing. So we welcome you to the this webcast is New Advances in Research for Recovery After SCI, and we're very excited about it, and we're excited to have you here. The webcast is going to have an online chat component, so anyone that's on the webcast can be chatting along the way. You can also ask questions, and Judy will be sure, as time allows, to get those questions in during the question and answer period. And we will also have this webcast archived on our website after the event, so you can access it anytime you want. And now I'm going to hand it over to Dr. Leslie Morse, and she is going to do introductions for us for this evening. Thank you, everyone. We're excited to have you.
So before we get started, I'd just like to mention that Caitlin Weiler is in the audience this evening. We're very fortunate to have her join us this evening. She is Miss Massachusetts International, and spinal cord injury is a very important topic for her. She is a very strong advocate for the cause, having had experience of a family member with a spinal cord injury. And so she is here tonight to volunteer and participate in the lecture series and hopefully get to know the community here in Boston. So please take a moment to stop by and say hello to her if you have the opportunity. So our first speaker is Dr. Jeff Macklis. He's from the Harvard Stem Cell Institute, professor of stem cell and regenerative biology at Harvard University, and professor of neurology and neurosurgery at Harvard Medical School. He's going to be discussing the basic circuitry that connects the brain to the spinal cord. He'll talk about the kind of injuries that are central to motor loss or loss of muscle strength after spinal cord injury, repair, regeneration, and or replacement of these neurons or elements of the circuitry linking the brain to the spinal cord injury by developmental and stem cell biology might provide useful real-world therapies for real people who have lost motor function in bowel and bladder control due to spinal cord injury. He will be highlighting a new set of approaches that his lab is using to pursue this goal. Dr. Macklis.
Reframe what Dr. Morse introduced, because unless I get a rousing, unanimous show of hands that you want me to dig deeply into molecular developmental neuroscience, I don't think you're all here to have me tell you about the fine details of the latest advances from my laboratory or laboratories I know. And I think what most people are here for, at least this is what I've been told, is to try to have me, I'm here for you guys, so to have me give a sort of broad overview of some things that go wrong and the circuitry and the parts of the nervous system that are really injured in spinal cord injury, and how we might think, and how laboratories around the world, my own included, are trying to change the long decree that spinal cord injury has no treatment or cure or improvement. Is that in fitting with people, what people would actually like? I was planning to do that in English rather than biologicalese or neurosciences. Is that in keeping what people, because I'm happy to do whatever, but I thought I would just do it in English. So what does this title mean? I put in this toward because I'm not going to try to sell you on anything or tell you that we're there. Spinal cord injury is a really tough problem. And what I thought I would do is tell you about some of the parts of the nervous system that are centrally affected by spinal cord injury and why it's been such a tough nut to crack. But then I'm going to tell you a little bit of optimism without giving you some golden wonderfulness, telling you that in the last decade or so, huge advances are happening that make me think that we're actually moving toward regenerating. That means making the nerve cells that have been injured, crushed, or cut to actually regrow, or repairing by replacing those nerve cells to essentially repair the circuitry that's gone wrong in another way. And we're getting toward that, and many of you know this term, corticospinal tract or corticospinal circuitry. All that means is we talk about the cerebral cortex of the brain. That's the highest part of the brain that we use for this conversation, for controlling our fine motor movements, for language, for thinking. And that brain, we all know, needs to connect essentially to our body. That's done, and I'm going to show you a little of how that actually works. That's done through long cables. And those cortico-spinal connections are the cortico-spinal nerve or neuron connections that are damaged in spinal cord injury. Okay, and I'll show you some pictures of this, but I think it's important to point out that the spinal cord is not some structure like a liver that we can sew it back together and it's right. It's more like a railway line. So corticospinal is like saying the Boston-New York Railroad. It's going from one place to another. And if we have a big injury in the tracks along the way, we need to connect those tracks together. And I'm going to try to tell you a little bit about how people are thinking about that. And the second part of my title is learning how these corticospinal neurons grow initially can help us to get them to regrow. And I think any of us in any of our jobs, professions, or places in life can think about the idea that many of us have cars or have had cars, and we know that if I just brought a car to somebody who never saw it and said, this doesn't work, can you fix it? We could change out a whole bunch of parts, and it would take a whole long time, and we'd never get there. But if I take it to somebody like Click and Clack, the Tappet brothers, who know how it's put together, then they can say, in that 87 Chevy, you know, they put the fuel line in this way. And the way we can fix it is to take these parts out, to put in a new fuel line, and to connect. So I'm just going to try to tell you a story in English that my lab and a number of others are trying to understand the genes and the molecules that build these cortico-spinal connections in the first place when we're all growing as embryos. And then we're trying to make adults, or not adults, but kids, adults, who already have those connections that are now crushed or cut or broken, to have them redevelop as if they're developing in an embryo, but to do it again in an adult. And you know, I officially have 15 minutes and then we have 10 minutes for questions, but I'm happy to take questions along the way, because if I lose somebody along the way, it's not doing any of us any good, okay? So just ask if, now that's the longest I'm going to spend on any slide. I have about 15 more slides, but most of them are just pictures. But that's sort of the basic story, that in spinal cord injury, the axons, the cables of the cortico-crossed spinal motor neurons, among other axons, sensory axons, bringing sensation in, they're damaged. And that loss of that connection, if you don't like the train metaphor, you can think of it as a telephone connection from telephone central station out to the various connections. Those cables have been damaged. They're not working. How do we rebuild them? How do we regrow them? And I think we're going to hear more about that as the time goes on. So Here is a human brain. Old drawing. This is that crazy complex cerebral cortex I was talking about. If we opened up the skull and looked at a human being, this cerebral cortex is essentially about as big as this tabletop in each of our brains, and it's folded up. And that's why it has all these folds and twists and turns. And in one area, just about there are these neurons that connect down to the spinal cord. And the rest of the brain might see and to figure out what that's seeing, figure out who that is, figure out what we want to do, figure out what action we want to take. But then the neurons sitting right here send those signals down through the rest of the brain as cables. They come down to the base of the brain. They come down through the brainstem. They come down and they cross, and that's why somebody who's had an injury on the left side of the brain, for example, has problems with the right side of the body. And these are happening both sides. They cross. They go down to the spinal cord, and they connect to a whole other set of nerve cells in the spinal cord that I'll show you that go out to the muscles. Is this making sense? Okay, so that's the cerebral cortex. And if I draw that on this guy, those connections are coming down, and here's the spinal cord. They're coming down into the spinal cord, and we all know that spinal cord goes about 2/3 of the way down our back. So, believe it or not, these nerve cells, and we usually think about cells being some tiny little round thing, these nerve cells have part of them essentially an arm. We call it an axon. And that arm, I'll show you a picture, can be astronomically long. It goes all the way down the spinal cord. From a teeny little cell that's about 1/10 the size of a human hair across. So those are millions and millions of cables that get injured. So how can we think about this? Well, I think everybody here can take a guess or knows what these cells are. These are red blood cells. They're like the simplest kind of cell. They're round. They float around. I hope I have no hematologist colleagues here who are going to get mad at me. But, you know, they're very important to us, but they're pretty simple. They have to hold on to oxygen. They have to give it up. If they go wrong, we can put in some new ones. We can give a transfusion. Problem is, here's a nerve cell. This is a simpler kind of nerve cell, or neuron, than the ones I've been telling you about. So it has this cell body that's about 1/10 or 1/5 the size of a human hair, so it's tiny, but it's pretty big by a neuroscientist point of view. It has all of these big branches. They're called dendrites. They're essentially antennas. That nerve cell is listening to about 10,000 connections on average each coming in. It's like central station of the telephone network. So it has to have all the exact right antenna branch dendrites, and then it sends something out that some of you might see me pointing out with this arrow, and that's that axon. And that axon, compared to this cell body, can go on the order of 10,000 times this far away. So what does that mean? Well, that means, I'll get to it actually in a moment, I'll point it out here, that if that nerve cell, that neuron, was as big as my fist, and it was a corticospinal neuron, it would send its arm, its axon, down the spinal cord, something like 5 or 6 miles. That's what we're dealing with. And it would do that. It wouldn't just send it randomly. It would send it out of this building. It would basically have Google Maps or MapQuest. That's what I use to get here. Because there are certain other cells in the body, in the brain, that tell it where to go. And it would know, because it knows how to read those signals, turn right out of the parking lot, go 300 meters, turn left, merge onto the highway, get off at exit 3, Go onto the Mass Pike. Go until exit 17 in Newton. Get off. Go up this street. Go up that street. Go into this office building. Find a room. And it would find a target about as big as that projection screen. That's pretty amazing. And that's all programmed by genes and molecules. The problem is that all happens when we're much smaller. And then once the nerve cell finds its target, then the body grows. So it actually doesn't have to grow those 6 or 7 miles to Newton. Is this making sense? It grows maybe half a mile, and then that mile extends. So the problem is, with spinal cord injury, we have to figure out a way to get those axons, either the ones that are already there and have been injured, or new ones, to find their way to Newton in a whole different territory. That's a big problem. But let me go back to this and just show you. So here's this human cerebral cortex again. The other complexity that I haven't told you about, and I know I'm telling you a whole bunch of negatives, but then I'm going to tell you the positives. If we looked in there and just sort of made a cartoon of it, this green and orange picture, along with those corticospinal motor neurons, there are literally thousands of other kinds of neurons crossing and twisting and turning and putting their axons. They all know where they want to go, but if you looked at it, it would look like all the roads and the traffic of Boston. Everybody's going in different directions. So we can't just make a new neuron, because it might not go to Newton. It might go to East Boston, and that might be a nice place, but it's not going to connect to the spinal cord. So we need to figure out not just how to make a nerve cell grow, but how to make the right kind grow, how to make it go to the right place and make the right connections. Now, one first bit of good news. The brain is actually wildly, amazingly cool. So we don't actually have to get the nerve cells to grow to exactly the right place. Because if we get slightly wrong ones to go somewhere and they're sort of a little bit off, the brain can figure out and use ones that are going slightly to the wrong place and sort of use them to make movements. And we all know that because we know people who have had injuries to a limb or have had something, a muscle that's been damaged, and we know that the brain can use imprecise connections. So that means trying to approach spinal cord injury, forgive me for saying this, but we don't need to get it perfect. And I hope nobody here will be offended, but all of my friends and colleagues with spinal cord injuries have told me they certainly wouldn't get offended at this. I think we can have a goal in this field of not aiming for perfection, not aiming for ballet and doing fine impressionist art, but rather to get useful function to real people. And that means that we can think about this problem of how do we get not to exactly the right table in the right room in Newton, but how do we sort of get our connections down the Mass Pike out to the suburban towns, and then let the brain again learn how to move and function. So that's what I'm going to tell you about. And I'm going to tell you about specifically that these corticospinal motor neurons that make us as humans do fine function, like playing the piano or writing with a pen or whatever, they exist in mice. And they exist in mice to do things like pick up a piece of food and manipulate it and bring it to the mouth. So it means we can study these neurons in mice, and it means that every molecule and gene that we and others have identified in mice, to my knowledge, also works in humans. That means that we can use this laboratory work. Some people might say, well, gee, you just want to fix spinal cord injuries in mice. That's not the point. The point is we want to fix them in mice so we can fix them in people. Is this making sense? Okay. So here are 3 of those corticospinal motor neurons, 1, 2, 3. They sit in their right place. These are the guys that are more complicated than the one that I showed you. Here are their antennas listening to 10,000 input connections. Here's another part of their antenna dendrites listening to a lot of other connections. And then they send their axons down here. And here's another view of the human brain. Here's the eyeballs up here and the back of the brain here. And here are those neurons sending, this is a cartoon, of course, sending those mile-long, not really mile-long, you know, it's a couple of feet long, but in my metaphor, mile-long neurons down through the brainstem. And then this is the spinal cord that's been enlarged. It's not really as big as half the brain. It's more like as big as my thumb around compared to my brain. But these axons are going down the spinal cord, and then they connect with those nerve cells that go out to the muscle. So this is the problem that I think I'm going to tell you about for just a few more minutes, and I think Dr. McKerracher is going to tell you about in a way of trying to get them to grow. And that's some of the problem that a lot of us who are looking towards corticospinal repair are interested in. So here's that neuron that sends its long axon. Now, it gets a little more complicated, but that's okay. Remember I told you that these guys send their axons into the spinal cord. Something has to tell the axons and each neuron whether it wants to be twiddling your thumb or moving your fingers or squeezing your hand or moving your arms, or whether it's moving your body, moving your legs. And I'm showing you that here by, there are ones that are shorter that go to your neck and control the arms, and there are ones that are longer that go all the way down the back. And everybody here knows that depending on the level of a spinal cord injury, that affects everything below the injury. And why is that? That's because it's not the injury to the spinal cord itself that's causing the problem in almost all cases. It's the injury to all these axons, all these cables that are going through. Does that distinction make sense? So we can't just look for some cells that will build a little bit of spinal cord there. That's not going to do us any good. We have to look for ways of getting these connections to make those long connections and grow. So here's the punchline without showing you the data. My lab has come up with ways of finding these neurons in mice as they're growing in embryos. At each of many stages, when they're reading those signals, getting their axons, growing them, and finding their targets. And we've been able to identify, using some methods that I'm not going to bore you with, the genes and the molecules that those neurons use when they're first building this whole system. It's like Click and Clack, the Tappet brothers, going into the Chevy factory and seeing how those cars are built. We've gotten the assembly instructions for these neurons. Does that make sense? And we're now trying, we'd love to rebuild this beautiful corticospinal circuitry like this mansion in Newport, Rhode Island, with all the precision I told you about. But I told you that we're not going to hold our standards up that high. So what we've identified is some basic framing rules. How to build a house, how to lay a foundation, how to put up the walls, how to put a roof in, how to hang a door. And our goal is we don't need to get to this beautiful mansion. We'd like to build a nice, functional corticospinal system to bring function back to folks with spinal cord injury. It's a pretty nice house. It's not as nice as the mansion. I don't think we're going to get perfect right away, but this might really bring us function. And I think my point is that, and there were a couple of slides in here, I guess in the revised version that I sent today that didn't show up here, that I'll just tell you the story. Spinal cord injury was first described somewhere between 1,700 and 3,000 years BC. In Egypt, in a doctor's papyrus, where he talked about battle injuries. And he declared, if you find a man, because these were all battle injuries, who cannot move his arms and legs, and cannot feel, then this is not to be treated or cured. Period. And he described a couple of cases. And about 120 years ago, a very famous neuroscientist, Spaniard, called Santiago Ramón y Cajal, said something very flowering like, after the development is ended, meaning after the embryo is born, the founts of growth dry up irrevocably, never to be restored. And that is why neurons cannot regenerate. But then in the next paragraph, he says, I leave this to the science of future generations to reverse this harsh decree. And I think in this past decade and in the coming decade and years, there's enough knowledge from hundreds of labs around the world that we're trying to actually reverse that harsh decree. And I would leave you with the thought that this developmental biology is, I think, going to lead in multiple ways to first small advances, then medium, then bigger medium, then larger, probably not perfect for a long time, advances in regenerating spinal cord injury. And I think that's all I have. And I think I'm about on time, and I'd be happy to take any questions. Yes? Here, you can use mine. Oh, okay. Great.
Thank you for that very useful introduction. I know that some of the people here have spinal cord injuries where all of the damage is in one place, and some of the people here who have spinal cord injuries have ischemic damage where there was damage from lack of oxygen in multiple places. What is the difference in terms of trying to get things to regrow between trying to ameliorate those two conditions?
Yeah, now because of time and because I didn't want to bore people, I left out hundreds of sort of devil in the detail parts. And I don't want to go into all those now because we only have 10 minutes or so. But the short answer to your question is, it's a really great question. Because you can imagine if I go back to that telephone cable metaphor, if there is a sharp cut, that's a much easier thing to think about splicing together. If there's a localized explosion, the equivalent of a severe traumatic injury, where the cables are disrupted, that many of them might be covered in rubble, and that's the case in human spinal cord injury, where we know there are things called glial scarring, so there's scarring around the area. There's all kinds of inflammatory response. One needs to figure out a way not only to reconnect, but also to get through all of that scarring and rubble. And then the case that you raise, where ischemia or lack of oxygen or blood flow in that metaphor is more like if there's damage across miles or different areas of the territory. That just means that even one local solution, even if we get through the rubble, isn't going to do it. And it may be that there's other tissue there, without being too negative, that one needs to have to even support those new cables. So ischemic damage that's broad and has led to loss of tissue, I think will be an even more difficult solution. Now that said, let me say the following positive thing. I don't think, as I've already said, that we need to rebuild all these cablings to get useful function. And there are ways to think about, rather than repairing what's already there, recruiting entirely new neurons, and that's something my lab is interested in doing, who haven't been exploded or damaged or covered in rubble, and have them be essentially like baby embryonic neurons. And just have them, it's the equivalent of saying, we can't fix this cabling, let's just lay a whole new line. And maybe lay a whole new line that goes around that ischemic area and takes advantage of the tissue that's not damaged. So on one hand, I'm telling you that it's probably harder for the regeneration, and on the other hand, I'm telling you that there are probably multiple workarounds. Is that an answer to your question?
Partly. Would the initial phase of that be giving someone the experience of having, they're still spinal cord injured, but it would just feel like it's lower down? That you could, as it were, push the area of damage down?
I think that's probably getting into a, you mean the experience of trying to fix it?
Yeah, that you don't try to fix all of it, you just try to give them a couple more inches.
No, I think it would probably be the idea of giving partial cabling, not as many phone lines as originally, but have them distributed around and have the brain learn to use things in imperfect but decent ways.
Thank you.
Are there other questions here? Yes, in the back. Yes, please.
I understand that people with recent injuries have more possibilities to benefit from these studies than people with old or longer time with the injury. Is that true?
I think this is an exceptionally smart and insightful question, and it's something that's of considerable interest in the field. And the scientists who work on this, and I think spinal cord injury physicians who work on this, make the difference between an acute or a new injury and a chronic or an old injury. And that scarring and tissue damage that I was just describing, in essence, and I know I'm going to run short of time, so I'm going to try to make my answers shorter, gets more established over long periods of time. So I think many people are trying to get approaches that might work first with acute injuries, and then maybe try to expand them, extend them, and make them better to work in chronic injuries. But you've highlighted a very important point, and a second point you've highlighted is not every spinal cord injury is the same. We all know that. You all know that. And that there are going to be ones that are incredibly complex, but relatively simpler. And there are going to be ones that are even more complex that are going to be really, really hard. And we probably can't come up with approaches that are going to be useful for everybody all at once. Is that an answer to your question?
Yeah, and I understand that the stem cell research right now is in the first phase, right?
Yes, I think, you know, that's I won't give a long answer on that. There are many, many, many different meanings of the term stem cell and many, many, many different types of research using stem cells. What I told you about here is called by some stem cell research, looking at the stem cells in the developing brain and how they turn into the right neurons. There are other kinds of stem cell research that think about taking cells and transplanting them in. Some of those make sense, ones that want to build new connections, and to my way of thinking, some of those don't make sense at all, trying to just put in some tissue locally in the spinal cord, because the point is to rebuild the cable connections. Now, the exception of that is there's some people trying to say, we don't need to rebuild the cabling, that's going to be too hard. Let's put in new computers down in the spinal cord, have them listen to the guys coming down from above, and have them send new connections, essentially making one long cable into two parts of a cable. So there are all kinds of creative workaround solutions that people are working on, but yes, we're at phase 1. There was a question, a fellow in the white t-shirt, and then, I'm sorry.
So you've been talking about neuroplasticity and kind of reusing connections that already exist. So labs like Redware Cortans that are looking at accelerating plasticity using different therapies and drugs, where would you say the breakdown is in promoting new tissue growth in damaged areas versus just trying to get the brain to work harder or smarter at rewiring the surviving tissue?
So I'm going to give you a very partial answer because I don't want to bore everybody else, because I think you're a biologist or you read the biology. So this raises a question that I can address to everybody, that even though there's a very large number of these corticospinal connections, or on the upside, a very large number of sensory connections coming up, and other motor connections coming down from other parts in the brain, very large numbers. It turns out, by a lot of studies in people, in mice, in other animals, that we only need something like 3 or 5 or 7% of them to function. We might not be the perfect ballerina. We might walk like I do, right? We might not be the perfect painter. We may draw stick figures like I do, with 3 or 5%. And the question that was just raised asked, one part of it is, what about if there's just 3 or 5% left? Can we get them to function better? And there's some research around the world that's trying to do that by having those few neurons have more effect. There's other research around the world where sometimes those cables are still there, but they've been stripped of their insulation, their so-called myelin. And there are approaches where people are putting cells in to try to rewrap those and get those few remaining axons to work. And then another that was just raised here is we don't necessarily need to use those same circuits that I told you about. Maybe for certain things like Reggie Edgerton, who's the advisor of this fellow Cortine in a prior life, there are other connections that go down the spinal cord that maybe if we just hijack them, we can get them to turn on certain responses that might just lead to holding up the body or maybe alternating stepping. Not perfect, but something. And I think there are hundreds of labs trying to take creative solutions into using the nervous system in ways that it's not initially built. Is that a partial answer to your question? Yeah?
Yeah, thank you.
There was, yes.
Sorry.
In the soon-to-be championship brewing sweatshirt.
Dr. Macklis,
Yes.
I'm sorry. We have a few questions on the webcast.
Oh, okay.
If I can jump in. And one is, are you familiar with chondroitinase ABC research for glial scarring?
The answer is yes, and we may hear more when I finally leave you. But you know, I can't in this amount of time tell you about all the avenues. When I told you just briefly about this glial scarring and the scar that forms at the damage site, there are multiple aspects of that. Some of it is certain inflammation cells come in and sort of set up roadblocks. Some of it is certain kinds of cells don't allow the cables to pass them. And some of it is that cells put out various chemicals into the environment. And that's what I was referring to as rubble. And some of that is chondroitin sulfate proteoglycan. Some of it is other stuff. And chondroitinase, ACE just means an enzyme that breaks it down. That's essentially a chemical, an enzyme, that can try to peel away and break down that rubble. And there are multiple other kinds of chemicals and inhibitors that I think we're going to hear about that make the cables not want to grow, and there are multiple molecular and chemical approaches to try to block those so that the axons can regrow. So that's the answer. Yeah.
Thank you. And one other question. I know there are many other questions that people would like to ask. We will circulate information on websites that will address these kinds of questions, and we'll get some information from you. So we'll send that out after the lecture, after the webcast. But one more question right now is there a lot of coordination and sharing of research between your labs and the labs around the country and around the world? And thank you for all you're doing for the spinal cord injury population.
The short answer to that is between many labs, there's immense collaboration and sharing. Not to talk so much about my lab, there are consortia, there are groups of labs that have been set up by the Christopher and Dana Reeve Foundation for Paralysis Research, where multiple labs will get funding, but, and this is actually a hitch that I think many of you would applaud, only if they all will come into the same room and tell everybody else what they're doing and share their reagents, share their results, and send people back and forth. We have a similar consortium that my lab is part of where we do the same thing. We get together every month and every lab tells every other lab everything we found out. And that, the goal of that is, 2 heads or 12 heads are better than one, and it advances everybody's work. And I think the real goal's got to be to bring something to this room in the future rather than just to advance our research, hopefully. But could I just answer the brief question that you had, and I'll try to be quick. Yes. Yeah, so the axon is really, to say it again, but now say it in another way, it is part of that cell. It can't live on its own in mammals. It can in some very, very simple organisms, but it can't in mammals. So after that axon is cut, the tissue below it disintegrates. But it's almost like wiring a house. There are some PVC cables, PVC pipes, through which those cables ran, and those sometimes don't go away fully. So depending on how long after the injury, if you're asking, can you take advantage of those connections down below, some of us think that we might be able to put the new cables through those tubes if it's soon after the injury. Longer and longer, getting to the woman's question about acute versus chronic, that tissue becomes severely disrupted frequently. And in some cases, if the injury is very severe, then everything is really totally disrupted. So it depends on the kind of injury.
Dr. Macklis, thank you so much for such a fascinating talk. I'm hopeful. I hope that you'll be able to come back and talk to us again soon about this. I know that there's a lot of interest in the room. Thank you. One quick additional housekeeping item. If you have parked in the garage here on site, please remember to validate your parking ticket at the front desk before you go down to the garage. If you go to the garage without validating it, you'll be charged full price, and we want to validate your parking this evening, so remember to do that. Okay, so a couple of issues. I'd just like to mention that Frank Reynolds was scheduled to speak this evening. He's co-founder of In Vivo Therapeutics, and he has asked us to give you his regrets that he could not be here to speak tonight. It is for the best of reasons. A few weeks ago, In Vivo Therapeutics received approval from FDA to begin the first human trials of biopolymer scaffolds in patients with new spinal cord injuries, and so they have a lot of work getting that trial up in running. So that's very exciting, but at the same time, sad that he couldn't be here. So hopefully also we can have him come back at another time. So I'm very pleased to introduce Dr. Lisa McKerracher. She's the founder of BioAxone Biosciences. BioAxone is a privately owned American corporation with a focus on bringing drugs to market for unmet medical needs. Specifically, they're working on Cethrin. It's a new therapeutic protein in phase 2 clinical development for the treatment of acute spinal cord injury, and she's going to speak on aspects of her work this evening. Thank you.
First, I want to thank the organizers for inviting me today, and really thank everybody that I know in the audience for having welcomed me to Massachusetts. And I think Angus, who's going to speak after, is going to talk a little bit about that. So I won't talk about that. But we know that everyone in this room has been touched in some way by spinal cord injury. And I'm going to give you an update on the research landscape, and in particular talk about my company's efforts to develop and commercialize a drug to treat spinal cord injury. But before I do that, I thought I'd just give you a little bit of background of how I got into this field, and what really makes me interested and challenged to really pursue the advancement of drugs to treat spinal cord injury. So personally, I had my own experience with spinal cord injury when my father fell off the mast of a sailboat into some water and some rocks. And I was about 10 years old and didn't really understand what was happening. Our cottage in Canada is on an island, so it took some time before the urgent care could get out there by boat. So it was quite traumatic. I remember it quite well to this day. I'm lucky because my father actually recovered. He has chronic pain resulting from it, but he's alive today and able to walk. But I've always had a love of biology, and I studied biology in university. I went on and did a PhD in cell biology. And it was really as a postdoctoral fellow that I became interested in spinal cord injury. But my scientific interest was when I started to work with Dr. Albert Aguayo at McGill University, who's really accredited with showing that neurons in the central nervous system have the ability to regenerate. Now, Dr. Macklis gave an excellent introduction to a lot of the things I'm going to be talking about, and he told you that it used to be thought that it was hopeless to even try and get neurons to regenerate. And what Dr. Aguayo had done is we know that neurons in the peripheral nervous system will regenerate. So if you cut your finger quite deeply, you may lose sensation for a time, but that nerve will regrow. And what Dr. Aguayo showed is that you could take a peripheral nerve and graft it into the central nervous system, and lo and behold, neurons from the central nervous system would regrow into that graft. So when I started as a postdoc in his lab, what I became really interested in was why is it that they do not naturally regenerate? And so after being a postdoc, I landed a faculty position at McGill University in Canada. And in order to pursue this, we started some biochemical experiments to try and purify molecules that were actually blocking regeneration. And we were the first group to purify what we call a growth inhibitory molecule. And on that top panel there, I show you actually quite an old experiment that was done in the early 90s. And what we did is we took one of those purified growth inhibitory molecules and we put it on the bead, which is that white dot. In today's term, we'd probably call this a nanotechnology or nano bead, but essentially we put the inhibitory proteins there. And then we put some neurons in tissue culture and watched what happens when they regenerate and they touch the bead. So the very first panel, called A, I don't know if you can see it, but this is the axon that Dr. Macklis was talking about. So this long projection, the fiber of communication, and this is one that's regenerating its axon in a dish. And essentially it looks, we call that a growth cone at the tip. It's kind of like a hand at the end of the stick. And those fingers, the filopodia, they seek signals in the environment to tell them which way to go. And you can see on panel B, one single filopodia, so one single finger touches that bead, and then in panel C, the whole axon will retract back. And what happens after injury in the spinal cord or in the central nervous system, we know that the axons will try to grow, but they touch so many of these growth inhibitory proteins that they became frustrated and they give up, and they actually have what we call a retraction bulb. So in the lower panel there, I'm just showing you a schematic of what's happening on the cell membrane, so on the sensors, and those green bars are representing receptors that are recognizing positive cues, and the red bars are negative cues. And since that time in the, essentially in the early 90s, discovering growth inhibitory molecules, we know that there are actually quite a few of these growth inhibitory molecules. And so when we thought about what would be the best way to overcome growth inhibition, it seemed like the best way would maybe not be to try and knock them out one by one, but to understand the mastermind or signaling. And what my laboratory discovered was that this protein called Rho was a key signaling molecule. And we started developing a drug to try and overcome the inhibition. So essentially the idea is by targeting Rho, what we're telling the growth cone is ignore those red signals or ignore the inhibitory ones and just seek out those positive cues. So actually we developed a compound based on a natural product. It was known that there was an enzyme made by bacteria that would inactivate Rho. And so we used that as the basis to make a drug, and using recombinant biology, made a drug that could inactivate Rho and reverse these negative signals that growth cones receive. Now, in order to commercialize or bring a drug forward to development, it actually takes quite a lot of money. So what we did is we started a company, and actually the company was originally started in Canada, and we founded it with the goal to develop drugs, this drug to treat spinal cord injury. So I think you can imagine it's actually quite a challenge for an academic scientist doing research in a lab to start a company and start doing drug development and following the regulatory path with FDA to run a clinical trial. I started the company actually in the year 2000, and that year Christopher Reeve gave a talk at the Society for Neuroscience meeting. It was a keynote talk. And he really actually motivated me because Christopher Reeve was not only an advocate for raising funds for spinal cord injury, but he also motivated scientists. And essentially he said, I've seen lots of experiments with rats having spinal cord injury running around in cages. I wish I were a rat. And please bring these discoveries to the clinic. So with BioAxone, we were able to do that. And we applied for regulatory approval after completing some safety studies and a lot of proof of principle in animals to take the drug forward to clinical trial. So Cethrin is the trademark name because it's a recombinant protein. It's not a chemical. It doesn't have a chemical name. In the lab, we originally called it BA210, so we just gave it a number. And what it is, it's a biologic drug. It's a first-in-class drug, which means it's a new chemical entity, and it targets this protein called Rho. So when we thought about how are you going to deliver this, because biologic drugs are proteins, and they can't be given as pills, we got a panel of experts together, and mostly neurosurgeons who advised us and said, why don't you give it at surgery? Because most patients have surgery after acute spinal cord injury. Actually, the statistics are in the United States, 90% of patients with a traumatic spinal cord injury will receive surgery for decompression and fixation of the spinal column. So we developed this surgical treatment, so it's an acute care. And the mechanism of action is quite well understood because I told you that we developed the drug from a natural product, and a lot of people had studied this enzyme. So we know it inactivates Rho, and through a lot of studies, not just our own, we know that it will promote regeneration of axons, not just in a dish, but we can show that in animal models as well. It's neuroprotective, and because Rho is expressed in all cells, it has some effects on other cells, such as on the monocytes, which are invading inflammatory cells and thought to do some damage after spinal cord injury. And so it will reduce the invasion of monocytes. So Cethrin actually has multiple positive effects, and a single application has a durable action. So that means we give it once, but the action of the drug to reverse Rho is sustained, and we know it's sustained for over a week, probably much longer. So with that, we went forward and we ran a multi-center clinical trial. The trial was held at 4 sites in Canada and 5 sites in the United States. and we recruited 48 patients. There were 32 patients with thoracic injury and 16 patients with cervical injury. Now, when we run a clinical trial with a new chemical entity, the whole goal is safety. So we were looking mainly whether the drug was safe and well tolerated, but of course, everyone wants to know, did it work? So of course, we were measuring some measures of efficacy as well. And I can tell you, I'm just going to show you results from the cervical patients who were treated. We saw interesting motor recovery in the cervical patients who were treated. In the thoracic patients, there was more variability in the noise. There was some interesting signal in sensory, but we would need to look at more patients to see if it was an interesting trend. In that graph that I show you on the left side, it's actually quite a complex graph, but where you see where it says weeks, those are the weeks we followed the patients out after treatment, so they were followed out for 52 weeks or a year. And then on the front, or on the x-axis as we call it, there's some numbers that are the doses. So we started off at a low dose and we went to higher doses. And then the height of the bar is the motor score, so the ASIA motor score. So most of you here may know what that is, but each muscle is scored on a measure from 0 to 5 bilaterally. So each neurological level has a score of 10. And so if you look at the very left of that graph, those black bars with the 0.3 milligram dose, that probably looks like a control would look like. But this was not a controlled clinical trial. It's what we call open label because it was a safety trial. So both the patients and the physicians knew they were receiving the drug. But what's interesting is if you look at the higher doses, the bars are higher, showing higher ASIA scores. And when you translate to what does that really mean for those patients, we had 8 patients with C4 injury in our clinical trial. And on average, that translates to recovering arm movements, or in some cases, patients recovered full upper body movement. What we saw of the cervical patients, 31% of the patients converted from motor sensory complete to recovering some motor movement. And I forgot to tell you that all of the patients recruited in our trial were the most severe spinal cord injury. They were all what we call ASIA A, or a complete motor sensory complete. So they were completely paralyzed below the site of the lesion, both for motor and sensory. So 31% of the patients converted to recovering some motor movement compared to 10% expected. So we think these clinical results are really quite promising. And what my company is doing now is moving forward to plan, to plan for a next clinical trial. So the regulatory process for drug approval, it's a long process because we have to work on the assumption that we don't know that the drug works until it's proven in a randomized placebo-controlled trial. So where are we is, Dr. Macklis also mentioned that usually we look at acute spinal cord injury, or drug development is often done for that because the bar is a little bit lower in the sense that the injury is new. We believe there's more potential for repair, and also you don't have the complications of chronic spinal cord injury where there could be some changes in muscle and bone. So in the last year, we've put together a drug development and clinical plan, and we're planning to carry out what we call a phase 2B study, which is a randomized placebo-controlled study. We're going to carry it out in the United States and Canada, and probably at the same centers and maybe expanded into some additional centers. On the subject of chronic spinal cord injury, which I know a lot of the people in the audience here are interested in, we're also moving forward with that. Now, I personally believe that for chronic spinal cord injury, we want to do a combination therapy because we know that there are multiple different things that we can do to improve outcome, at least in animal models. And then when we combine them, we get better results. And one of the things that BioAxone is doing right now, we're collaborating with Dr. Wise Young at Rutgers University to combine Cethrin with umbilical cord blood cell therapy that he's developing. And I'm really excited about this because some of you in the audience may know that he's running clinical trials in China, but he's planning to begin those clinical trials in the United States. And what it takes to be able to combine two different and new compounds is that you must finish your safety trials first before you can combine them. So the individual compound has to be shown to be safe before you can start combining. And we've already completed that step with Cethrin, and he's working towards completing that step with his cell-based therapy. So once that part is completed, then we can combine them and begin clinical trials to treat chronic spinal cord injury. Well, we're early in that process. We're just at the stage of doing the preclinical proof of concept. So we have to demonstrate in animal models that this idea we have is really going to work, and also carry out combined safety studies before we can submit an IND or an investigation of a new drug for clinical study to the FDA. But I think this is a really, really exciting and new area, and I'm really looking forward to launching some additional clinical studies. Well, I'll just go back. So right now I'd say we'd like to move faster, and what's really blocking us, I hate to say, is partly financial. And one of the largest roadblocks in the field of spinal cord injury is actually a lack of understanding of the indication and the potential for new drug development. And I think this is something that everyone in this room can really help with, because people need to be educated on what the potential is. In the pharmaceutical industry, generally the larger companies are focused on the very large markets and large disease indications with lots of patients. And spinal cord injury is considered an orphan indication because, while it's a very important and it's an unmet need, there are not as many patients as there are in other types of diseases. And so when I go around talking to venture capitalists, raising money for my company in the clinical trial, they really don't understand the indication. And they don't understand that a very small neurological change, if you had a 1 or 2 level neurological improvement for a patient with a C4 injury that could now move their arms, this is tremendously important for quality of life. And this is the kind of education that I think is necessary so that people in general understand what improvements can do and what the potential for repair is. Because actually a very small amount of repair could make a tremendous difference for quality of life. So my last slide here says cures for spinal cord injury are achievable. And again, just looking at what are the different blocks in the process. And I'd like to just mention a little bit what's going on in other fields of medicine in general. First, there's been some really amazing advances in spinal cord injury research at the basic level. And we have a lot of understanding of different targets and different potential ways to repair the spinal cord. So we heard one of the questions on chondroitinase, and there are many different types of strategies. But right now we're sitting in the situation with not enough clinical trials going on, because to get drugs approved, we need to test lots of things to find out what's the best. And the only way to do that is to stimulate translation of basic research into clinical trials. So on the left, I have you there, I show you the process of going from basic research, showing it works, preclinical efficacy in animals. The next step in order to develop a drug or a new chemical entity is CMC, stands for chemistry, manufacturing, control. You have to be able to scale up and make the drug in a way where the quality control is there. So you have to make sure it's reproducible, and you can absolutely verify the potency and activity of the drug. And then after that, you can do safety studies in animals before doing the clinical trial. Now, traditionally, basic research is typically done in academic environments. I think the most innovative research is done in the universities. And these are funded by NIH or not-for-profit. And traditionally, clinical trials are usually done by companies because of the expense of running a clinical trial. And there's this in-between, which is making the drug and scaling up, which is actually very difficult to fund in this risk-averse environment, venture capitalists are less likely to fund in that in-between level. And what we're seeing in other fields are the not-for-profits coming up and taking up and helping in this area. So, for example, there's been some amazing developments done by some of the biotech companies in Boston. One of the examples is Vertex, which had a drug approved for cystic fibrosis. They had a collaboration with the Cystic Fibrosis Foundation that had put in lots of money into the program. And I think this is a model we need to see in spinal cord injury, is to share the risk and share the reward to move things forward. So the bottom line is I think this is a really exciting time, and I think there's a lot of different targets out there, and hopefully we're going to see combination therapies. In the 17 years since Christopher Reeve was first injured, there have been an incredible number of potential targets to treat spinal cord injury. And with some of them moving forward in the pipeline, I think we're going to see some exciting times. And I'm looking forward to coming back and telling you about our clinical studies that we hope to get underway soon. So thank you very much.
So we'll go ahead and take a few questions from the audience, and then from the webcast, we'll move on to our last speaker. Questions?
Hi. Do you know how in the chronic stage of the clinical trials with Dr. Weisjung, do you know where the site would be, the surgical site, if it is chronic? Can you address that?
We're not far enough in the planning to do. We're really planning at the preclinical stage right now, but likely it would be a multi-center trial, but that's really not been planned yet.
Thank you.
I'd like to pronounce your name correctly, Dr. McKerracher.
Very close.
Thank you for your work in this field. It's very important to us. My question is, the collaboration that you're having with Dr. Young, is your collaboration not also in China, or could you tell us why Dr. Young would go to China? Would that have something to do with our government putting up roadblocks?
I don't really want to, speak for Dr. Young. I've heard him explain why he's there several times. In part, it had to do with when the Chinese gymnast had her accident, and he talked to her and promised he would bring treatments to China because she was afraid that when she went back home, she wouldn't get medical care. So I've heard him say that it was a promise he made to her. I don't remember her name. Also in part because the not-for-profit foundation in Hong Kong is funding the studies. So it's partly, I think, where the funding source is coming from. But he is planning to bring it here as well. So I know he's working towards clinical trials in the United States with the same types of therapies.
I think we have a question from the webcast.
Thank you. Can you speak at all to how it would work in combo with the chronic animal trials with Cethrin and cord blood? Do you have any thoughts on that at this point?
The reason why I think they would have synergistic actions, which is mean that 1 and 1 wouldn't be 2, they would be 3, is because of different mechanisms of action. And one of the problems with any cell-based therapy, even given an acute spinal cord injury, is that the cells are usually transplanted later because there's such a huge inflammatory environment, it kills the cells right away. So any clinical trial that's been done with cell-based therapy in acute spinal cord injury, usually they wait 10 days, 2 weeks. And by that time, some of the damage is irreversible. And so what we're thinking is if we combine them, we could give an earlier treatment. And one of the reasons is because of the neuroprotective action of Cethrin that should help protect also the cells that are being transplanted and also stimulate the endogenous neurons to regenerate and respond to those cells. That's why we think that the two would be very powerful together.
Great, thank you. And I think we can take one more question.
Mine's almost more political, but I'm like, why do lab rats have more rights than we do? How come we can't volunteer to become part of these studies and get into these preclinical things, maybe before they've gone through all of the full FDA jumps and hoops and ladders and whatnot, because I feel like our government is what's keeping us from having a cure.
No, I think what's keeping it is that there aren't enough clinical trials, because then people could be volunteering to go into the clinical trials. And I really do support the regulatory process, because people forget that there could be drugs that are not safe. And we know some horror stories that have happened in the past, like thalidomide, that caused a lot of birth defects and children born without arms. And that's because of a lack in the process of protecting patients.
Well, thalidomide passed all of the government tests that existed at the time, and it was pulled off the market by the manufacturers when it was discovered to be a problem, despite the fact that it was still useful for treating injuries for people who were not potentially getting pregnant.
Well,
but I'm saying, even with the horror stories, I'd rather be able to take the chance.
You know, I can't say yes or no to that. I do agree with the regulatory process. I think we need to get more academic scientists taking the risk to take their great discovery forward instead of just going on to another great discovery. That's really what I think needs to happen in this field.
I want to have like your lab rat.
Okay, thank you very much for your questions. Thank you so much, Dr. McKerracher. And again, we look forward to you coming back and updating us soon. Please remember to fill out your surveys, give us feedback, give us any ideas on topics that you'd like to hear more about, and gives me great pleasure to introduce our final speaker, Mr. Angus McQuilken. He is the Vice President for Communications and Marketing at the Massachusetts Life Sciences Center. This is a quasi-public agency of the Commonwealth of Massachusetts. The center's mission is to create jobs in the life sciences and support vital scientific research that will improve the human condition.
All right. I have one of these, but I do need, how do I get my slides up? No problem. What's that? So it's forward. So it's forward. How do I go back? Good evening. Thank you very much for the opportunity to be here and say a few words. Again, my name is Angus McQuilken. I'm here representing the Massachusetts Life Sciences Center. I'm not a scientist. I don't intend to pretend to be one. You have two really amazing scientists tonight who have been able to give you some insight into the science and answer a lot of great questions about the science. My agency plays a different role, and I'll explain to you tonight what that role is, some of the investments that we've made that relate to spinal cord injury. First, to give you a sense about why we're doing this work here in Massachusetts. Massachusetts, as many of you know, is a global leader in the life sciences. And when I say life sciences, that's inclusive of biotechnology, pharmaceuticals, medical devices, diagnostics, bioinformatics. We have strengths in all of these industry sectors. And that has allowed Massachusetts to emerge as very much a global leader in this field. And what makes us, what positions us to be a global leader in the life sciences? Well, we have world-class academic institutions, some of which you're familiar with, MITs and Harvards and University of Massachusetts facilities being good examples. We have a talented workforce, best educated workforce in the United States, with a lot of talent in fields related to the life sciences. Life sciences is a very important part of our economy. 95,000 people in Massachusetts wake up every morning and go and work in the life sciences. And we've seen a lot of growth in this field, 50% biopharma job growth in just the past decade. And we also have government. playing a role that is not necessarily being played in other states through the passage in 2008 of a 10-year, $1 billion initiative to invest in the acceleration of growth in the life sciences, really with two goals in mind. One, to create jobs, and the other is to accelerate science and scientific advancement toward the development of new treatments and therapies and cures that'll improve the human condition. So what we do at the Life Sciences Center, we are the agency that's implementing that 10-year, $1 billion initiative. So we are the agency that makes the decisions about how those resources will be used to accomplish those two major objectives, creating jobs and advancing good science. So we serve as the hub of the life sciences supercluster, as we call it in Massachusetts, and we make investments. And I'm going to tell you a bit tonight about some of the investments that we've made that relate to spinal cord injury. Really, we make investments in 4 areas. We invest in scientific research to keep the pipeline of innovation strong. We invest in companies directly to support those companies' growth and development. We invest in workforce development because talent is the #1 reason why the life sciences prosper in Massachusetts. And we invest in capital projects, construction projects that relate to the life sciences. So this is how the initiative that you're all paying for, assuming you live in Massachusetts, this is how the initiative is structured. Again, over 10 years, it's $1 billion, which sounds like a pretty big investment. But to bring one therapeutic to market, as Lisa will tell you, often costs more than $1 billion. So we have to use these resources wisely and leverage private investment as a way to increase the impact we can have in this important field. Half of the resources of the Life Sciences Initiative in Massachusetts are actually for construction projects, capital projects. And to give you an example of one that, that relates to spinal cord injury, we invested $10 million in a new facility at the Marine Biological Laboratory in Woods Hole toward the creation of a new center on for regenerative medicine. They're doing some really fascinating work there. For example, they're researching how is it that the xenopus, which is an African frog, has the capacity through the gifts of Mother Nature to regenerate the lens in its eye when that lens is damaged. That's what they do at the Marine Biological Laboratory. They research marine organisms and develop treatments for humans based on what they can learn from the natural processes of these marine organisms. And one of the projects they have underway there is research into the lamprey. The lamprey is a creature that can regenerate its spinal cord when it's injured. So they are researching what can we learn from that creature from the ocean that we can translate into treatments, therapies, and cures that can be applied to human health. So half of the initiative's resources are for capital projects. So we just invested last week $95 million in a new therapeutic facility, scientific research facility at UMass Amherst, for example. A quarter of the initiative's resources are in the form of tax incentives through which we encourage life sciences companies to grow and add jobs in Massachusetts. And a quarter of the initiative's resources are in what we call our investment fund. And those are the resources we can use for grants to support scientific research, workforce development, and early stage funding for promising companies. And this is what we've done so far. We've invested $461 million from that initiative. That's allowed us to leverage about $1.2 billion in third-party investment to amplify the impact of the investments that we're making at the center. That's a 3x multiplier on the public dollars that we're investing. So we really have 3 functions at the Life Sciences Center. First off, we're a funding agency. We fund things. Secondly, we serve as the state's marketing arm to attract companies in the life sciences to Massachusetts. And the third role we play is as a convener. And I'll tell you about how these different functions of the Life Sciences Center relate to spinal cord injury. One thing that we do at the Life Sciences Center is we support promising early stage companies. Now, Frank Reynolds was not able to join us tonight. Frank Reynolds, as you heard from his statement, is the founder of In Vivo Therapeutics. One of the first companies that the Life Sciences Center chose to fund through our accelerator program, which provides loan financing to early stage companies, is In Vivo Therapeutics. In Vivo is developing groundbreaking technology to treat paralyzed individuals with spinal cord injuries. And they're doing it through the use of biodegradable polymer scaffolding. It's a very innovative technology, as was mentioned in Frank's statement. They recently received, just this past April, FDA approval for the very first human trials using this type of technology to treat spinal cord injury. We gave this company a loan in 2009 of $500,000, a modest investment by the state. The company has been so successful in raising private funding that just 2 years later, they were able to repay that loan with interest after raising more than $30 million in private capital. So there is private money out there to support research and promising technologies in spinal cord injury. And this company, which had just a few employees when we funded them, I think they had 5 when we funded them, they now have 50 employees. And they're located in Kendall Square, and they have those people there every single day working to develop this technology and bring it to the market. And that's really one thing I want to mention about the role of the Life Sciences Center. We support research, we fund research, but our goal is to bring that research to the market so it can reach patients. Our goal is commercialization. So in everything that we fund, that's what we're looking to do, is see that technology commercialized so it can reach the patient. So these are a couple of photos that you'll see here. We have the repayment when In Vivo Therapeutics raised that money and repaid their loan to the state, and then a ribbon cutting at their new headquarters there in Kendall Square. By the way, the woman in the red suit is our president and CEO, Susan Wyndham Bannister. Right behind her arm, it's not a great picture of him, is a guy named Justin Fowler. Some of you may know him. He won the 2012 Ironman Triathlon, and In Vivo Therapeutics is one of his corporate sponsors. He's on my list of inspirational people. A second company that we have funded through our accelerator program is called Myomo. Myomo is a medical technology company. They are using medical robotics. So this is another important and growing field in Massachusetts, is medical robotics. So they're developing prosthetic technology, powered bracing devices that restore an individual's mobility. And they're looking to use their platform technology of medical robotics that restore mobility in a variety of ways to treat people with a variety of physical conditions. This company won Mass TLC's Product of the Year award in 2011. We gave them a $750,000 loan just 2 years ago, and they're one of the promising companies that is now in our portfolio. So we're a funding agency. That's one thing that we do. We fund capital projects that relate to the life sciences, like the work at the Marine Biological Laboratory. We fund companies. We fund scientific research. We also serve as the state's marketing arm to attract promising companies to Massachusetts. This is something we've had a lot of success at. Massachusetts is viewed now as the global leader in this field, thanks in large part to the investments that are being made through this initiative. And that's allowed us to attract a lot of companies to Massachusetts, including some that are bringing promising technologies to the market that relate to spinal cord injury. So last year, AdvaMed, which is the National Industry Trade Association for companies in the medical device field, had their annual convention in Boston. And this was a great opportunity for us to showcase Massachusetts as a great place to do business for the companies that are developing these sorts of technologies. And at that convention, we had the opportunity to announce, and you'll see Governor Patrick here in this picture, the president and CEO of our agency. We had the opportunity to announce that Argo Medical Technologies, a company that is based in Israel, had decided to establish their first U.S. headquarters, and had decided to establish it here in Massachusetts. They've actually opened that facility in, I believe it's in Marlborough. This company makes a system called ReWalk. ReWalk is a robotic exoskeleton that allows individuals with lower limb paralysis to get up and walk again. And at this convention, I had the opportunity to organize an event where Sergeant Teresa Hannigan, who you see here pictured using the ReWalk system, who is paralyzed from the waist down due to a degenerative condition, she was able to use this system, which she has been trained on using at a rehab hospital in the Bronx. She was able to use this system to not just get up and walk a few steps on that stage, but she then got down from the stage and spent about a half hour walking around that convention hall with a bunch of TV cameras following her, and everyone she walked by was just amazed at this new technology. So these are the types of technology that are coming to Massachusetts, being further developed here and brought to the market. This technology, by the way, is approved now for use in rehab hospitals, and it is pending FDA approval for individuals' use in their own homes. So this will be an important technology to keep an eye on going forward, ReWalk. And another company that we attracted to Massachusetts was BioAxone. Now, Lisa, as she mentioned, she was hanging around in Florida where the weather's nice and LeBron James plays basketball and they don't know how to count votes after an election. Florida. But we convinced her that Massachusetts was the right place to bring her company. And we were so pleased. We first met, Lisa and I first met at the Bio International Convention, which was also in Boston last year. So AdvaMed is for medical devices and diagnostics. The Bio Convention is for biotech and pharmaceutical companies. And Lisa and I met in the Massachusetts Pavilion. We had a conversation about why Massachusetts is a great place to do business. A few months later, BioAxone arrived. And this picture is an event that we had at the Cambridge Innovation Center to celebrate this company's arrival here in Massachusetts. So since she's already explained the science of Cethrin, I won't even try to do that with you tonight. But I will say, because she's very modest about her company, that they were selected by Windover Conferences as one of the top 10 neuroscience companies for partnership throughout the world. Is that right? Excellent. Now, we also, as I had mentioned, serve as a convener, by which I mean we bring different elements of the life sciences ecosystem in Massachusetts together to collaborate. And we do that in a variety of ways. We do it by promoting public-private partnership, by bringing the public sector together with the private sector, academia, and industry to work together and collaborate. And we do that by creating what we call pre-competitive consortia. I'm going to tell you a little bit more about what that is. But those are efforts to get different companies, often competitors in industry, to work together in new ways toward the goal of achieving scientific breakthroughs and bringing new treatments and therapies and cures to the market. So an example of a pre-competitive consortia that we formed just this past year is the Massachusetts Neuroscience Consortium. We announced this at the BioConvention, the same one where I met Lisa, in the Massachusetts Pavilion. It is a new model for development of technologies in neuroscience. What we have done is convince 7 of the leading global companies that invest in neuroscience research, companies such as Abbott, Biogen IDEC, you'll see them listed here, EMD Serono, Janssen Research, which is part of Johnson & Johnson, Merck, Pfizer, and Sanovian. We got these 7 companies around a table, 7 companies that spend most of their time competing with each other, and said to them, you're all spending a lot of your own money on the same research in your own labs. And a lot of that money is going to waste. If you were to collaborate, so you heard earlier about the models that exist for academic collaboration where they're trading information about their work and their progress. What we said to them is, what if you pooled your money together? And through the Life Sciences Center, we ran a program where we could fund neuroscience research on all of your behalves, preclinical, early research. You would then all have access to the results, and you could then use your money to move those promising candidates for drug development and other technologies forward. What if we did it that way? And what we were able to do is get these 7 companies that had really never worked together before to agree to each contribute in the first year $250,000 for a total pool of $1.75 million to support neuroscience research, an area where there is a need for more funding, as evidenced by the fact that in the first round of that program, we received 97 applications from researchers across the spectrum of neuroscience seeking funding through this initiative. And we will be announcing the first round of awardees under that program next month. So what is the Neuroscience Consortium? As I say, it's a pioneering new model, really an unprecedented model that we want to make work in the field of neuroscience and may then extend to other fields like cancer research and other fields where collaboration between companies in the early phases of research could yield great results, but also a lot of economies of scale. So what's in it for industry? Well, they get leverage on each other's investments, and they get access. They all get access to the results of that research instead of having to invest in it individually and separately in their own companies. And what's in it for academic institutions? Well, they get funding that's hard to find. So there's another source of research funding available through this consortium. They get leverage, and they get access. In this case, they get access to industry, to the resources, the tools, the equipment that's often not always available in an academic setting, but is often available in an industry setting that is necessary for this research to move forward. And another role we play as a convener is to bring together communities of people who care about certain areas of health and health technology to learn from each other. And Dr. Eric Ruby is here, and he is a terrific human being, and many of you may know him. I've had the opportunity to work with him for several years now on the organization of Spinal Cord Injury Awareness Day. So one of the things we do at the Life Sciences Center is support efforts like Dr. Ruby's effort at the State House each year to raise awareness. So each year, the Life Sciences Center is there to sponsor Spinal Cord Injury Awareness Day, and our president and CEO, still wearing that same red suit, she owns other clothes, by the way, just want to mention that, is there every year to give an update on what we're funding and the progress being made through these investments to address spinal cord injury. So thank you very much for the opportunity to say a few words.
We can take a couple questions.
If you ask me any scientific questions, I'm going to say, Lisa, thank you for being here.
Thank you. One of the problems which was discussed by one of the earlier presenters was the problem of getting funding for taking a therapeutic idea from the point of being academic research to the point where you could commercialize it. There's a couple of intermediate steps that are very, very difficult to fund. Since it's so difficult to get those funded, is anybody in your organization trying to make available funding for that sort of a bridge thing so that we can offer something that people will not find somewhere else?
Sure. That's a very good question. What we try to do at the center, given we have some substantial resources, but they're still limited resources, is we try to identify gaps in the life cycle of health technology development. So one gap we identified early on was this gap in early stage financing, right? The so-called valley of death, where promising companies with promising technologies never get off the ground because they can't get some early funding that allows them to get to the point where they might be able to appeal to venture capital firms, angel investors, and raise more money. So we look for gaps along the pipeline of innovation, and you've identified one of those gaps. As I mentioned, it can cost well more than $1 billion and take well longer than 15 years to bring a new therapeutic to the market. That costs a lot of money. We're never going to be able to, at the Life Sciences Center, provide the resources that are necessary to get to that stage. What we can do is promote collaboration that will allow for that work to move forward in a more cost-effective way. And the Neuroscience Consortium is an example of that for early research. One thing that we're working to do and are having some good traction on is international collaboration. No one country can solve these problems. And if we think we can, we're making a big mistake. There are brilliant people all over the world. And to solve these problems, we need to allow these best minds around the world to collaborate with one another. That's how we're going to get there. And so we're serving as a platform for collaboration. We have a new program we just launched called IPAP, which is an online portal where companies from Massachusetts and then from all over the world can post profiles about themselves so that they can find each other and partner on their research, whether that be early stage or later stage. Think of it as online dating for life sciences companies. So those are some examples. It's a huge challenge. The model's broken for bringing expensive therapeutics to market. There's a lot of people trying to think through how to fix it. There's no easy solution, but it is a big, big challenge that's ahead of us. Thank you for the question.
Any other questions?
Terrific. Thank you all very much.
I think we have one more, if
one?
that's okay.
Sure.
Do you provide loans or strictly, do you provide grants or strictly loans?
We provide a mix. So
Okay.
the tax incentives that we award to companies that are growing in Massachusetts are grants, essentially, in that they reduce the company's tax liability, but they don't need to be repaid. The scientific research grants that we provide are also grants. But for the early stage companies, what we provide is loan financing. Give them the resources they need to get up, get moving, access private capital, then they can pay back that loan. So the only loan program we run is the program for early stage companies. I should mention, how many students do we have in the room? Are there some students here? Okay, so I mentioned workforce development is also one of our areas of investment. Just for the students in the room, please know that we run an internship program that provides funding to pay students and recent college graduates to go work at life sciences companies. We've placed more than 1,100 interns at more than 300 companies across the state. So anybody who's here and might be interested in such a program, please come talk to me afterwards and I'll tell you how to access those resources.
And just additionally, how do companies go about applying for a grant?
So we run the accelerator program, which again is a loan program, twice a year. And it's all of our programs are competitive. So we open up a solicitation. Companies that want to compete for funding can apply. We run a very rigorous review process that includes peer review, a scientific advisory board. So for example, in a typical round of the accelerator program, we will receive about 40 applications. We'll probably end up funding the 4 most promising of those companies. But what you would need to do is keep an eye out for us to announce solicitations, which you can get on our email list in order to do that via our website, which is masslifesciences.com. So that's how you can find out about it. And you apply actually via our website. It's all online.
Okay, thank you.
Great.
Thank you again for a very interesting talk. And I'd like to thank everybody for coming tonight. I hope to see you all at the next Knowledge in Motion lecture series, and stay in tune for more information on that. Thank you.